Okay, good afternoon, participant. Welcome back for the day 2 of the Student Research Symposium. Now we'll be going forward with our plenary talk number 6, that is by the Professor Shailja Singh from JNU, Jawaharlal Nehru University, Delhi. I'll just give you a short brief about our speaker. Professor Singh has been extensively involved in the fundamental research on biology of human parasites, malarial and leishmania, and has shown a great potential in the translation research towards the drug discovery and development. Her work on a nanomedicine-based artesunate with surface-coated iron oxide nanoparticles has enhanced the anti-malarial potential in P. falciparum and its multi-drug resistant strain. And we hope that she will be definitely sharing this particular work with our members. And also this work has been published in eBiomedicine. It is the Lancet publication journal and this particular work has got the Gandhian Young Technology Innovation, that is GYTI award in 2018 and was conferred by the President of India. So ma'am, you are most welcome. And now the all platform is all yours. So kindly share with your presentation.
So thanks a lot for this invitation, and it's always a pleasure to interact with the young minds and exchange of ideas. And then, of course, whatever research we are doing, it is due to the innovative ideas which our students they come up, and then we work with each other in a team to achieve something for the society. So I will start with some of the background from my research, how the pore-forming toxins and perforins we are using it. And then in my later part of my talk, I am going to go towards the iron nanoparticle fortification of artesunate, which again was a new idea, which was like a new idea from our side, and we thought, can we improve our artesunate therapy, which is there for malaria treatment? Can we do something about it? And we got the success, and it was a real, again, a pleasure to have that success in terms of, because it was going towards the translation towards the society. So to the student, still I really would like to convince you that there is a need for malaria research. We keep on saying that the malaria elimination programs are in place, and then we are cutting down on malaria incidents. And we also say that the nearby, like our neighbor countries, like they are also, like now China is saying that they will be eliminating malaria, and they are declaring that they are now free of malaria. Then Sri Lanka already declared themselves to be malaria free. But for India, because as I say, we have the very much diverse geography, and we have so much forest area around us and everything. So that itself are the main burdens for us to have the malaria elimination program implications. So in that case, we know that the deaths related to malaria, specifically to children less than 5 years, are really, we cannot cut it down. So there is again a need. But again, if you see this graph, which is at the right side, that we are putting a lot of effort to just get rid of this disease. Just we want to declare that then we are malaria free now. Our India is malaria free. That's what we would like to do in 2030. But by that time, what are the needs and what are the challenges? So the challenges, as I said, are multiple. The mosquito, which are the carriers for this disease, malaria disease, from one human to another. So there are multiple species of mosquito. They have different habitats of mosquito. So their habitats are also changing. For example, we are developing, let's say, insecticide. People are doing some good research. So they are developing novel, the chemistry people are developing a novel insecticide, because the old insecticides are not very good for the mosquitoes, because they are becoming resistant. But then the new ideas are coming, no, you just genetically engineered the mosquito, make the mosquito which are devoid of parasite, because if they will not carry the parasite, they are not going to transmit from one individual to another. So how they would be devoid of the parasite? You either the mosquito should be good in their immune health, so they can kill the parasite whenever they are getting that parasite. But the second thing, they can also be like, we should have a strong insecticides where we can kill the blood feeding mosquito. If not all, we can kill them. Specifically, let's say if you are treating me as an individual and then mosquito is biting me and mosquito is dying, and the mosquito which is not biting me will not die. So we are selectively only killing those mosquito which are vector for our disease, like if the vector for our disease like malaria, dengue, chikungunya or so. So these are the effort. Then the let's say there are Wolbachia stories, like every individual like me, you and everyone, we have the microflora in our body, like we carry nice bacteria, good bacteria, helpful bacteria, fungus in our they are the in our gut flora. Similarly, mosquito also, they carry virus, bacteria and the fungus. So can you have some type of bacteria which will be there in the mosquito midgut, and that fungus will kill the parasite inside the mosquito? Some way those approaches also already been there in the field and people are trying, and that's what we want to handle the vector story, like a mosquito story or the vector which are responsible for the transmitting of one disease from one individual to another. So you come up with a novel idea, say I have developed this genetically engineered mosquitoes, you free in the environment, when they will mate with the non-genetically engineered mosquito, then the progeny will die. So everything you can try, and people are doing that. Secondly, we are also working on that we have identified a gene which will if we delete those gene or inhibit that gene, the sperm of the mosquitoes are not very healthy, they are not motile. And if the mosquito sperm will not be healthy and motile, they will not produce more progeny. So in that way, we can control the mosquito as well. So that way, one way, but you can also handle the disease. It's the more transmission part. But the disease part is that you have to, if you have got the malaria infection, you need to treat the individual, specifically the children who are really suffering. There is a cerebral malaria problem, because if the parasitemia goes very high, it goes to the brain, and then you get the coma. Even children with the malaria parasite infection, especially P. falciparum, they comatize. There is a problem in the liver, there is the problem with the kidney, everything, like a multiple organ failure also happens. So with that, I just want to convince that still we all need to do research. We need to find out the new ways to tackle this disease, whether it's the vector side or towards the parasite side. So we would like to have the transmission block, meaning if I am getting infection, I should not transmit it to another individual. Second, if I have got the infection, treat me, so I should be cured for that disease too. So if you see the whole life cycle, we need to understand how we get the infection. So it's very it looks very complicated, the life cycle, but just to tell you in a very simple word, that whenever the female Anopheles mosquito, which is a full of parasite, because the so for that requirement, that infected mosquito, whenever it bites to a healthy human, what it does, it takes the blood meal, but it also injects the parasite. Then the parasite from the muscle, it goes to the liver cells. So our liver cells are the first home for the parasite. So parasite go to liver, they enter to the liver cell, they grow, and then finally you see there is a rupture of liver cells, and then the parasite come to our bloodstream. So, and then when it comes to bloodstream, now their second home is the red blood cell. So we have multiple red blood cells in our blood. So those red blood cells, when infected by human malaria parasite, then the parasite forms a very nice ring-like structure. We say ring stage parasite, then trophozoite, they grow. They form a multinucleated structure that we call the Shailja. Then they break open the red blood cell. Again, multiple merozoites come out and then they infect the new red blood cell. So that way it goes. Now, what happens when I get the fever? When is the point I get the fever if I have a malaria parasite inside my body? When it is there in the liver, we don't get any symptoms, because that is not the symptomatic clinical symptomatic condition for the malaria. But whenever from liver it comes to blood, then it causes the fever. And then the malaria parasite, when we are infected in our blood, there is a fever, and then when the parasite is coming out, we get the chills. So if you remember, if you are from any endemic area, you must be remembering that the fever comes very late at night, very high fever, and that time we also get the chills, because our red blood cells are just rupturing, and then we get the all symptoms of malaria. But at that time, what happens? Some of the parasite, they just decide, okay, I have to make some sexual stage of malaria parasite. So then they turn into male gametocytes. Some of the parasite, they commit themselves to become a female gametocytes. So the male and female gametocytes are formed in our human body, and at that time when we have the male and female gametocyte also in our infected blood, then there is again a bite. If at all we are getting mosquito is biting us, the male and female gametocyte are also going to mosquito, and then inside the mosquito, then male and female, they fuse, they form the zygote, then ookinete, and then finally infection to mosquito. Then that infection increases, and finally you get the sporozoite stage to the salivary gland of mosquito. And then once again that mosquito will bite to some individual, so that will transfer the sporozoite to healthy individual. So you can see now you have a plethora of things. Where do you want to treat to block the malaria infection? You want to treat in the liver stage when I have an infection, but I don't know because it is inside my liver. I am not getting any symptoms, but yes, I can treat them because I have a vaccine against liver stage. So I'm already vaccinated. As soon as I am getting the liver stage infection, it will be clear. So the all vaccine approach is made towards the liver stage of malaria. Then you come to the blood stage, because blood stage you get all the symptoms. So whatever drug is being developed, generally been developed towards the blood stage, because I get the fever, I need to kill the parasite. So artesunate, that is artemisinin, and chloroquine. These are the hydrochloroquine. These are the drugs which are being used for treatment of malaria, is basically the blood stage. But if I want to really block the transmission also, so in that case, what am I going to do? I'm going to also kill the gametocytes present in my blood. So not only going to treat human for malaria parasite, but also going to treat the gametocyte. Now, if you'll come and tell me, any medical doctor will say, okay, take this medicine and that will kill the gametocyte. I'm not going to take it. That's the compliance of the field. So you have to give the therapy in such a way that you are giving for the cure of that person, that it will kill the malaria parasite, but you also need to have the composition which will kill the gametocyte. So there comes the symptomatic malaria transmissible. But if I am asymptomatic and you are going to give me the gametocyte drug, I am not going to take, meaning not me, the compliance is very, very poor. So in that way, all drug designing, everything is being done in such a way. So I said liver stage, then the blood stage, then the gametocyte stage, and then the mosquito stage too. Because if you are making some genetically modified mosquito and other thing, you are freeing them at the field, then you are taking care to the mosquito stage. Then you also develop some new insecticides. So those are the very important things to have your target places. The very much important thing is that even you can have something in the body, and then when the mosquito is taking it, you are killing something inside the mosquito, like that we call the endothelial. So we are also developing some endothelial. So when I was doing some this kind of a work, we came across to one protein like perforin-like protein, and that perforin-like protein, as I say, we are saying it's a perforin-like, meaning it has a performing ability, but it's just not perforin. Perforins are there in human, you know that, like cytotoxic T lymphocytes, they have the perforin-like molecule. Whenever they see an enemy like bacteria and other thing, they just secrete this perforin-like protein, and then they kill the pathogen. Similarly, malaria parasite also has this like protein, that is a perforin-like protein, and there are 5, 1, 2, 3, 4, 5, because it's a family of protein. One family contains 5 proteins, and those 5 proteins are expressed at the different stages, like a liver stage, then the blood stage, then the gametocyte stage, and also in the mosquito stage. So we thought, why not to take this protein, which is there in the all stages, and then target that protein. So we can target by one drug or one compound. We can target the liver stage, we can target the blood stage, we can target the sexual stage, we can target the mosquito stage. So it would be a good idea, but the challenge was to have the common domain present in all these family members. So the family members are 1, 2, 3, 4, 5, and the common thing present was the MAC domain, which I have here represented in the gray color. So you know there is a middle central domain, and this middle central domain, every member have this. And this is what is this domain? This domain is only the business end of the protein, because the business comes from here in terms of forming the pore. So if a protein has this MAC domain, it will bind to the membrane, and then it will help in the oligomerization. And once it is oligomerized, it will insert to the membrane and form the pore. So the pore formation by this family members are governed or mediated by this MAC domain, MSCPF domain. So we thought, why not to target this MSCPF domain, because it is present in all family members. But before that, any research, first you need to really convince the field, convince yourself that this protein is there in the malaria parasite, where it is located, what is its function. This you can do. So just to convince you, like in green staining, you can see this is a malaria parasite Shailja stage, and the green staining represents the perforin-like protein. You can see very nice punctas are there associated with the parasite, that is blue in color, that represents the DNA content of the parasite. And then you also see a very nice ring-like staining, which is representing the host membrane, because the parasite is inside the red blood cell. So the protein is being made by the parasite, and then it is being secreted to the surface of the RBC, because it wants to rupture the red blood cell to come out when it is mature. Like for example, the parasite, if at all, it's just, if you will lock the parasite inside the host, that is human red blood cell, it will not be more infectious. It would be just taken care by the immune system, and it will be cleared by the host. However, the parasite has its own arsenal, own protein, which helps it to come out. But parasite does not want to come out when it is immature, because if it will come immaturely out, it will not be able to make the next infection. So to have that cycle in a proper time-bound mature level, the parasite will make the protein in a very just-in-time. And just-in-time action of the protein will help the parasite to be running on its life cycle. So in this case, we also found that very late stage, when the parasites are about to come, that time these perforins are expressed. Similarly in gametocytes, because gametocytes are also male and female, if they are stuck inside the red blood cell, they are not going to fuse, they are not going to make the zygote. So they also need to come out from their host to have the further fusion to make the zygote and the formation of ookinete in the mosquito stage. So there also perforin has a role. Then in the sporozoite, when the sporozoites are coming out of the mosquito and going to our liver cells, so it has to just open up the liver cell membrane to enter. There also the role of perforins are coming. So that's what, having those thoughts, we decided to first look at where are they expressed. We found, yes, they are expressed towards the membrane and also associated with the parasite. But again, to prove that if at all I say that these perforins are forming the pores, they are pore forming like protein. So I need to give you the proof. So what we did, we made the recombinant protein, we incubated the red blood cell with the human red blood cell with the perforin like protein recombinantly. We are the only one in this whole globe, like I can say the malaria community, that we can make this perforin like protein. We purified, we incubated with the human red blood cell, and this is the atomic force microscopy pictures, where you can see these are the, we are just seeing the surface of the red blood cell, and these are the pre-pore structures. So which is very clear that we are seeing the pore formation when we are incubating with the red blood cell. The perforins are incubated with the red blood cell, and then we can also measure what is the depth of the pores, what is the assembly of the pore, what is the diameter of the pore. So that's what we convince the field. If we are forming a pore, of course, what will happen? Red blood cells, if they are attacked by the perforins, there would be a discharge of hemoglobin. So hemoglobins will be out. You can measure the hemoglobin by spectrophotometer. The standard 405 absorbance will give you the stain. So you can see, as soon as I add more and more perforins, there is a more and more pore formation, bigger pores, and the hemoglobins are coming out, and then I am in the supernatant. I'm seeing the hemoglobins. If at all I'm measuring the hemoglobin, I can see, okay, my perforins are working against the human red blood cell. So to really give you a sense that these perforins are helping parasite to exit, and this is a calcium regulated manner, because the perforin, they don't work alone. In the presence of calcium only, they form the pore formation. They do the pore formation. So here, this is a very interesting live cell video microscopy. So we always say the parasite is inside the red blood cell, it come out, it goes inside, and that's what it cycles. But what if I am going to see that under the microscope? Time-lapse video microscopy will really give me the events and the real proof to my research that yes, it is happening. And when it is happening, what happens to calcium, what happens to perforin? So you can see here, when I just loaded the Shailja stage parasite with the calcium dye, calcium sensitive dye, meaning it will, the green color will represent the dynamics of calcium. So when it is more green color, meaning calcium is surging, there's a trigger in the calcium, inside the calcium, inside the cell, this calcium ion, and then if I am seeing some event. So I'll just run this movie, and you can see there is a blast, just like a blast, and then there was a flux, the calcium flux, and then the merozoites are now out, and they will attack to the new cells. So with that, I thought of, let's chelate the calcium and ask the question, if we have calcium, yes, perforin will come out and it will stain the membrane. But if we have chelated the calcium, there should not be perforin now, because their function, their secretion, everything is a calcium dependent. So it was a very simple experiment where we chelated the calcium by a compound like BAPTA-AM, and then we asked the question, where are the perforins? So in a very late stage, as I said, the green dot again represents, this is the three-dimensional rendered image of Shailja, where we are staining the perforins. So if we have enough calcium, there is a surge of calcium, the perforin go and bound to the host membrane. It decorates, because there then it will tear the membrane and the parasite will come out. But if we chelate the inside the calcium of a parasite, we don't see any decoration of perforin on the host cell membrane, and the parasite will be inside, locked up inside the red blood cell, and we don't see a increase in the infection. So with this, we also ask, if at all perforins are now out, let's say we found that they are on the membrane and they are helping to parasite to go out from the red blood cell. Let's say if I want to go out the room, from this room, there would be a gate, and that gate is being formed by the perforin-like protein, so I can go out. But if I enter another room, for entry also I need something to have a smaller pore for me to go inside. So we also ask the question whether could see, yes, they are there at the junction. When the parasite is attaching to red blood cell, they are there at the junction, and then the junction opens up and the parasite goes inside, and at that junction point there is a perforin-like protein. And which one? There is another perforin, that perforin-like protein 2, because I said there are 5, 1, 2, 3, 4, 5. One is present in liver stage, one is also present in the blood stage, 2 is present in the blood stage and gametocyte stage. So they are distributed, but the function are almost the same. So that way, but now to prove it, yes, invasion is also being regulated through calcium and the perforin. We now focus more towards the invasion story. So in that invasion story, if here is the new red blood cell. So as I said, there would be exit of the parasite, then the parasite will enter to another cell, and the entry point will also be held by the perforin, which is been discharged by the parasite. So you can see here they very nice. Then rupture, merozoites are out. Then it attacked to a new red blood cell. That it went inside the new red blood cell. Anything will come to a cell. Cell will get disturbed, right? So the red blood cell also got some echinocytosis process to happen to accommodate the new parasites coming. And you know, this is happening to our human body because we have the blood. And if at all I have an infection, malaria will enter to my new red blood cell. So the new red blood cell will just try to accommodate, and then the infection will fall. There also, I said the perforin has a role. So block the perforin, you will block the entry also. That's what we could show, how you will block by antibody, because if I'm saying that this could be the vaccine candidate, so if I have an antibody to perforin, it will not allow the new parasite to enter. And if at all I have a drug, it will not allow my new parasite to come out. So that was the approach. With the for the entry process, we thought of using the antibodies. So we made the protein, we made the antibody, and we could show if we put the antibody, there is no calcium increase. So you can see here, there is a recombinant protein, and if you add the antibody, recombinant protein will be neutralized with the antibody, and there is no pore formation in the human red blood cell. So the antibody is effective, and then if at all it is effective, it will block the invasion process. But the problem was not only the perforin helping the parasite, but these perforins, because they have the pore-forming ability, they are toxic also. You know, when we get the malaria infection, we get there is endothelial dysfunction because the parasites are there along with the red blood cell in our blood capillaries, and the blood capillaries are lined through the endothelial cells. So what happens if the parasites are attaching to endothelial cells and they are in the vicinity, and then they are secreting the perforin? Maybe these perforins are also damaging my capillaries. So that was the question we asked. So again, when we took the perforin, like protein from the parasite, and we took human endothelial primary cells, and we incubated the recombinant protein with the human endothelial red blood cell, human endothelial cells, we could see the endothelial cells are not in a good shape. There are dysfunctions are being noted through the morphological examination. So we saw a blebbing of our endothelial cells, human endothelial cell. We also saw there is an increase in calcium, because everything, the human cells or any other pathogens, they are more towards the regulatory mechanism is through the calcium ion, cyclic AMP, GMP, and then coordinated function of cyclic AMP, GMP, and all. So we also see the calcium. However, I would really like to show you this movie where I have taken the human endothelial cell, I have taken the perforin, and then you can see how it's damaging the cells. So you can see there is a blebbing, like a cells are now started responding towards the perforin, and then there are bleb formation, and then those after all this blebbing and all, now the cells are really damaged. So how are they damaged? You can always see the marker. Everybody knows that in bioscience we ask what kind of a marker is it. Endothelial cells are dying through apoptosis, very known function, necroptosis, apoptosis and necrotosis both, necroapoptosis, anoikis, what kind of a cell death. Even we would like to know the cell death is, if cells are dying due to apoptosis, are they dying due to necrosis, are they dying towards the apoptosis and necrosis, because the response of our human cell, of the death response and the repercussion of the cell death also affects our immune system. Because if any cells are dying towards due to the necrosis, they will cause more inflammation. So that For those purpose also we need to really understand, and it's a basic science research also we would like to love to know what kind of a cell death is this. As a scientist, anybody will ask. So you can say that annexin, we got also there were annexin positive, then we use the JC1, this is mitochondrial dysfunction. So in endothelial, when we add the perforin-like protein from plasmodium, there is a loss of membrane, there is a loss of membrane potential of mitochondria. Then to see apoptosis and all, again, is a very famous dye, that is propadium iodide, which tells you there's a DNA fragmentation or not, integrity of the cell membrane, and then also the calcium. So we also did that, and you can see how the cells, they, as I said, they respond, they bleb, and then finally they die due to the necroptosis. So there is more and more calcium are getting up, and then when the calcium is getting up, it's the cells, and then you can see the blebbing is also now stained, and then they are PI positive, meaning membranes are compromised, and the PI propadium iodide dye, which represents the DNA fragmentation, has entered to the cell, and now the whole endothelial cells are dead when if they come into contact to the human malaria perforin. So then doing the HMGB assays, this is nothing. You can always read about it, that it tells you the cells are dying through apoptosis or necroptosis. It just defines whether it's a necrosis or the apoptosis. So we could see it is a necrosis process through the. Then we also look for the. So these are the story with the endothelial cells, right? But again, I am saying all our red blood cells are not infected. Some of the red blood cell, which we say the bystander red blood cell, in some red blood cell, parasite is inside, it is growing, and then it will come out, it will infect some of the new red blood cell. So parasitemia is, let's say, 5%, meaning out of 100 cells, only the 5 red blood cells will be infected. What about the other 95 red blood cells which are there in the blood flow? So we thought maybe these perforins which are being out from one parasitized red blood cell, they also might in fact disrupt the other bystander red blood cell, which are nothing to do with the parasite. They are just flowing in the vicinity, and if at all it will these perforin will rupture those red blood cells, there is always a chance of that you have a less hematocrit. And in case of malaria, we always feel that there is an anemia. So why anybody will feel the anemia when you have the parasite in the blood stage in your body? It is only the 4%, 5% of your red blood cells are infected. So we thought, no, it's just not the 5% of the total red blood cell, but the other red blood cells which are not been infected by malaria parasite, they might also be interacting to the some of the protein being discharged by the malaria parasite, and they might be affected. So having that thought in the mind, we checked, we again added the perforin-like protein to human red blood cell. We could see there is a nice green color coming up. They are more crenated, echinocytosis process. They are nothing to do with the infection, but just seeing the perforin-like protein coming from the another infected red blood cell, they could see that they are annexin positive, meaning they are also in a senescence stage. And then again, you can see the movie is a human red blood cell immediately drawn from my blood only, I think. And then we added the perforin, and you can see all are becoming green because we have just in the media we have added the fluo4am. Then you can see they are now bulgy, they are like a swollen, and then some of them will just rupture as well. So with that, now all this story I could tell you that perforins are really important. They are dangerous. They are making dysfunction our endothelial cell. They are also targeting our human normal red blood cell. They are helping parasite to enter. They are helping parasite to exit. But what to do? You have to have something to target it, and to target it, we look for the bioinformatic help. So in silico analysis we did, because the mouse malaria perforin structure came by that time. So we also modeled our human malaria parasite perforin, and then you can see all the members. They have this the MAC domain, which is represented in the red color, and then there's an alternate helical structure which goes inside the red blood cell. With this information, we designed some molecules actually, and those molecules then we did some computer guided approach. We took, we thought whether that is interacting to perforin or not. If at all these molecules are interacting to perforin, what are they blocking to? Are they blocking the self-assembly of the perforin, so they will not assemble to form the pore, or they are just like a cement, they are blocking the pore? So we think they are blocking the assembly of the protein, and I have the proof for this. So you can see this, like they are interacting, these molecules which we designed, they are interacting to two monomer interface. So if they are there in the interface of that interaction point, so the many monomers will not come close and they will not form the oligomer. If there is no oligomer formation, there will be no pore formation. So that way they work. So we proved it again when we add first thing to show that the perforins are interacting to this compound which we designed. So yes, they are interacting. You can see this is the SPR data. So, and then by other cellular thermal shift assay, like you take the protein, you take the compound, you give the temperature condition, and if the protein is protected, meaning they are binding to the ligand, the small molecule. If they will not bound, then the protein will be degraded. Those approach is very well known nature method protocols you can see, and that through SPR. After that, if I can see my compounds are interacting to perforin-like protein, they should block what they should block. They should block the lysis, they should block the calcium formation, they should block everything what I showed to you like in my last 6-7 slides. So in that case, you can see, yes, whenever I put the atomic force microscopy, there is no pore formation because they will not allow the oligomerization, how the pores will be formed on the red blood cell. Then with all this, there should be blocking the entry and invasion. If I'm using this anti-perforin molecule, so yes, I can see they are blocking the growth and they are also blocking the invasion. They are blocking the exit, which we call the egress. So perforins are not in the action because of this compound. Then not only they are blocking the blood stage, they are also blocking the sexual stage, we can say, because if they will not come out, there will not be fusion in the zygote formation. How you are going to see the gametocytes are coming out? There is a very standard assay, the exflagellation. So whenever the male gametocyte you take under the microscope, you activate them through the xanthonuric acid, which is very similar to mosquito level. You see then they start exflagellating. When they exflagellate, at that time, if you add the perforin-like protein inhibitor, you don't see the exflagellation. So with that, we concluded that they are not coming out from the red blood cell. Then there is a block in invasion, there is a block in egress, then there is a block in fusion. And there is also block in the liver stage malaria parasite, because I said for liver stage also the parasite needs to go inside the liver cell. So it will not go to the inside the liver cell because the perforins are required and you have blocked the perforin. So with this, yes, we could show this sporozoid growth is blocked and everything is okay. Then these compounds should protect the human red blood cell. Yes, they are protecting. You can see this is control in the lower panel. They are really good shape. And though perforins are there, but if I add my inhibitor, they are neutralizing the perforins. Then we could show, yes, they are also blocking the annexin coming out by the other signatures of Raman spectra, by using the Raman spectra of our peaks, which represents the apoptosis. We could show there is no apoptosis in human red blood cell when we treat our human red blood cell with perforin along with the compound. And then we again saw that the red blood cells are also now protected. And finally, and then with that we could show, yes, finally, when we design this compound, these are not only anti-malarial because they block the entry and exit, but they are also good in terms of saving our endothelial cells, saving our human red blood cells. So anemia problem or any endothelial dysfunction problem. So these are the from basic research how we have come up with a new drug discovery platform and how we have developed this new compound. Now we are testing them in the animals, and once we test them in animals, we are now planning to go ahead with testing them in maybe phase 1 and 2. We will think over it. So with this, I also would like to tell you our iron nanoparticle story, because there you can see that how not only having the basic research, but having having the already existed therapy, how our novel ideas or innovative ideas can help improving them. So this is the example of that paper which I thought of sharing with you. So just a second. So why is it not coming? Do I need to share again?
No, no, ma'am, you don't need to share again. Just make this slide full screen.
Okay, thank you. So here it is. Can you see now?
Yes, ma'am, it's visible.
So here the idea was very simple. You know, the artemisinin, we know this is an anti-malarial, well documented. Everyone, the therapy is acceptable to all across the world, because whenever you have the malaria episode, and if at all it is a falciparum, the artesunate is going to be the mainstay therapy for you to get rid of your malaria symptoms. So artesunate, no need to tell you, it's the same story to convince you. This artemisinin was earlier been discovered by this Chinese lady, what she again got the Nobel Prize for this drug. So this is a magical drug. Meaning for malaria, it's like a for falciparum treatment. This is the drug anybody should be knowing it for. Earlier it was a chloroquine. So the chloroquine was, we know that parasite have developed the resistance against the chloroquine. So the only therapy which is now in the treatment regimen and also been acceptable by WHO is the artemisinin therapy. So yes, so artesunate, what it is, chemical structure, you know, and then as I said, this is not only for malaria, but it is also been used for the cancer, now any chronic diseases, virus, you just name it, this wonder drug is now being tried for many other diseases also. So, but I want to bring all your notice to this peroxide bond, because this is the main mechanism of action of this compound artesunate. Why artesunate kills the parasite? Because if at all, because this compound has an endoperoxide bond here, and in the presence of Fe2 plus, it forms the free radicals, and the free radical formation is the mechanism of action of this compound. So now the question is, what is the problem with the artemisinin? That if at all you will provide more iron, it would be more active in terms of free radical formation, and you can improve the therapy. That was the only idea we had. And to go ahead with this, what we did was, like, again, it's more elaborated that this is artemisinin. There are the derivatives, sodium artesunate, meaning we have the more soluble form of artemisinin, and but again the bond is there. And then you can see here like dihydroartemisinin, which is a active metabolite that also has that bond. So whatever the form is, if at all you have this, you are going to see the effect, because this forms the ROS, the reactive oxygen species, in the presence of iron. So with this, what our approach was, and just to give you again more idea that this artemisinin is not only for malaria, but it is also anti-allergic, anti-tumor, anti-cancer, anti-angiogenic, antifungal, anti-protozoan, anti-helminthes, antivirus, whatever it is. The mechanism comes through the ROS formation, and the ROS is in the presence of iron. So with this, again, every year there is an increase in the this molecule. Why it got the Nobel Prize? Because everyone is trying it for the different disease, and every year the publications are increasing for its novel function for different diseases. So what we thought, for if at all this is the only drug for malaria, and in case of malaria parasite, when it gets activated in the food vacuole, because it gets the free or the heme bound iron, and then in that case it gets the like a free radicals has been formed, and that's what, and this happens in the food vacuole of malaria parasite. That is the mechanism of action of this compound artesunate. So we first wanted to see whether if at all, if I'm seeing there is a free radical formation, if I put iron, and if I don't put iron, I chelate iron, what happens? Is there any more ROS being generated? And it is well received and well documented by other system, but we also tried in our system. So when we put iron, of course, there is more ROS formation because the mechanism of action is being held by the iron. With this kind of background information, what we did, we talked to our chemistry collaborator, we asked them, can you make a nanoparticle which is now made up with the iron nanoparticle, and it should give us the iron when in some regulated manner. And that regulation was pH, because as I told, in my malaria parasite, there is a food vacuole. The point of action is at the food vacuole. There the pH is low. So I wanted them to make a nanoparticle in such a way that at the lower pH only the iron should be released. Because if the iron would be released in advance, maybe my artemisinin, which I am combining it with, will not be active at the time when I want them to be active. So with that, they made this by the simple method, like taking FeCl2 and 2FeCl3, and that for coating they have taken the compound, and finally with that kind of formulation they could make that nanoparticles. Nanoparticles characterization, you guys must be knowing better, that you can also, of course, you can do the powder XRD spectra. You have some other UV-based method to characterize your nanoparticle. Once they made the nanoparticle, now they also wanted to see this iron nanoparticle, what is the size, went for the elemental composition analysis, then by SEM, each particle, what is the size, if it is real nano or not. So 10 nanometer was the size they could calculate. And then the main aim for our biological experiments, the pH responsiveness. So I asked them to do the pH range, pH 5, pH 6, and pH 7.4, because I know in my blood stream the pH is 7, where I don't want my artemisinin to be active, but when it entered to the parasite, it should be active at the pH 5, pH 6.5 when it's in the cytosol. So that level, and what could they show? At pH 5 only these iron nanoparticles are releasing the iron. So that was very clear from their experiments. And then I could also then ask whether this nanoparticle have any toxicity to other cells, and we did this experiment. So just the iron nanoparticle alone was not toxic much, because it should be working with the artesunate, right? So in that case, we could show, yeah, they are not that much toxic. But once we combine it with the artesunate, so artesunate would get help from the iron, and that's what now it can be more active to kill the parasite, and that's what we could show here. Artesunate alone was, of course, it is a good drug, and there would be some iron inside the parasite, which is artesunate is using. But when we fortified it with the iron nanoparticle, the action is much pronounced, and the effect is much pronounced, and we do see there's a good. But then again, we needed to show whether this iron nanoparticle which we made, it goes inside the parasite or not, and specifically to food vacuole, because there I want them to release the iron. So you can see that here, like the black dot, it is a hemojoin, which is a marker for me to see the food vacuole. And in the food vacuole, near vicinity to food vacuole, that is black dot, I see the green color, and the green color is our iron nanoparticle FITC conjugated, just to see them, where are they. So we could see, yes, they are near the food vacuole. So they are in the right place to release the iron. So with this, again, if at all they are iron nanoparticle fortified artesunate, there will be more ROS. I showed you to the other cells, but in case of malaria or parasite also, you can see more green. More green because we are now looking towards the ROS. ROS, we are looking to the one dye, that is DCFDA dye, so it binds to the free radicals and then it fluoresces. That's what you can find it in the literature. So once there is more ROS, there will be more DNA damage, and that DNA damage would be only for the parasite. So looking at the more damage to the parasite, we could see through the COMET assay, yes, the parasites are dying due to that more DNA damage. Macromolecules are damaged. Macromolecule कौन कौन से होते हैं? The one macromolecules are, if at all, the major ones we say protein, DNA, RNA. So DNA, I could show that they are damaged. Then we also look for the protein. So the protein macromolecule alkylation, if the protein is damaged, we can see they are alkylated. So by DNPH staining, we can show that the protein in presence of iron and artesunate, the parasite proteins are alkylated. So that's what the proteostasis mechanism works, and the parasite they die. Then we went towards the animal, and we also fortified the artesunate along with the nanoparticle, and we treated the animal which are suffering from malaria, and we could show that even the 10 times less artesunate was similarly active when we just fortified with the iron nanoparticle. And iron nanoparticle alone was not doing anything, no toxicity, but when artesunate alone was killing the parasite. But when we combined the artesunate along with the iron nanoparticle, even the artesunate was 1/10 of the concentration which is generally being used. That 60 mg per kg is the standard dose we give, but when we give even the 6 mg per kg and then we add the iron nanoparticle, we do see a good clearance of the malaria parasite from the mouse model. So it's the animal data. So with this, we also look for some other toxicity. I said the other organs are they get affected: brain, liver, kidney, spleen, and they were okay. And so again, I want to say that with this work, what we could show that the that artesunate therapy, which is being limited due to some short span of life, and then the cost is also. With that, what we can do that our nanoparticle, iron nanoparticle fortified artesunate can be a good solution. Now I'm working with AIIMS Ayurveda Center, and what we are doing, we are taking the iron bhasm. Basically, they didn't know that knowledge exists. Whenever they come up with the malaria infection, anyone comes to the clinic, Ayurveda clinic, for malaria infection treatment or for anemia treatment, they give the law bhasm. And that law bhasm is nothing but iron nanoparticle. So I thought of talking to them, and I said that if we can work together, and our artemisinin, it's artemisinin, artesunate treatment should be given with the iron bhasm, and if at all that is our knowledge can help them to treat malaria in a better. So with this, I just want to say that the summary, what our knowledge we have developed, that the mechanism is known. Artemisinin is known. It forms the ROS, but the mechanism says that we need iron. So there is again a two-level, the iron nanoparticle formation. So combine the 3-4 knowledge, interdisciplinary science to have a better outcome for your research. So with that, I really would like to thank. I hope I have not crossed the time, and I would like to have the question.
Thank you, ma'am, for the nice session. Audience, if you have any query, please unmute yourself and ask to a question our speaker.
Thank you,
Ma'am,
Professor Singh.
Yes, please.
Thank you, Professor Singh, for your talk on.
Yeah, you are a bit. Can you be a bit loud? Yeah.
Okay. I think now it's okay.
Yes, I can hear you. Others can also. Yeah. May I know who are you? Your name, please.
This is Sanjay.
Okay. Yes, please. Yeah.
Actually, my question is related to the cost-effective treatment of the malaria. Actually, and you are basically propose the iron nanoparticles, or it's called iron.
Yeah.
So, how does it cost-effective? the
Yeah.
existing
So
soil?
see what I showed you at the beginning. The formation of this iron nanoparticle are very simple one-step process. No, FeCl2 and FeCl3, and along with the like a coating material, you can make the iron nanoparticle. That's what I have learned. That it's very easy, less time-consuming, and very simple method to do. And the starting materials are also not very expensive. But as in the end of my talk, I said that law bhasma already exists, but nobody knows that law bhasma from Ayurveda is the nanoparticle. So what I am proposing it, if at all we cannot make a new formulation with it, whatever I could show, but the principle is there, right? So can we combine the artemisinin drug along with the Ayurveda formulation of law, and then can it be the cost effective one? That's what the calculation should be. Yeah.
And then what about the basically the time limit for the treatment? Actually, that's an amazing concern. Sometimes malaria persons are suffered from the weak.
I totally agree. So, according to me, both the therapies are there, right? The artesunate therapy is there. We don't have to go for FDA approval, anything. Every ethical approval is there. For law bhasma, everything is there. Now it is the clinician, and we have generated the knowledge. Now it is the clinician. That's what I was asking them. Now you should find it out at least phase 1, phase 2, and phase 3 type of trial, where you just show that if you are giving this drug along with this drug, can it help? So those things, if anybody can show, I think it should help everyone. Yeah, I do agree it is a time-consuming process and all, but we all need to meet, all need to talk about in such a way that our knowledge and their knowledge can it be combined to have a better thing. Yeah.
Thank you, Professor Singh. So
Hello, ma'am. It was a very wonderful talk. The way you have explained it was, even though we are from electronic background, we got a lot into it. So I have a general. So I have a general question, ma'am.
Yeah.
You mentioned
What's your
in one
name?
slide.
You are which year electronics student?
Ma'am, I'm first year PhD.
Okay, first year
IIT Indore.
PhD. Okay, yes,
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 ma'am.
yes. Yeah.
So you have mentioned in one of your slides that you asked your chemistry collaborators to ensure that the iron nanoparticle is safe for all the cells. So, what is the there are many cells in the body, and it can be anywhere in the body, from what I assume. So,
Yeah.
how are we ensuring that it is safe for all the parts of the body? It's not
Yes,
just
yes. So
iron.
yeah, it's a very genuine concern. But I told you, when we have anemia, we are given to, we are also given the iron. Like you take him up. Basically, iron is for our to have more hemoglobin, and then our red blood cell. Like to treat malaria, iron therapy is one. But anything higher dose can be toxic. So my point was, I'm not saying that it will not go to another cell, but the concentration which we are using along with the artesunate, that is not high enough for not to give to any human. That was the only concern. If I mean the toxicity, meaning it is not toxic at the dose where we are using it with the artesunate, but rather it will help, because in case of anemia, if I'm getting the iron, my more and more red blood cell will come up. The children who are suffering from malaria, like in Africa and all, they are so anemic. So give them iron nanoparticle or lobhasma or anything. In one way, their blood cell will be more boosted, as well as they will be anti-malarial if you are combining with the artesunate. So I'm saying for artesunate combination only 3 days, but iron nanoparticle you can give even for longer days. They are not toxic.
Okay.
They will go to the cell, they will go to the human red cell progenitor cells, and they will help the more red cell to come out from our bone marrow. Yeah.
Okay, ma'am. Thank you so much.
Any query from audience? If there is no further query, once again, thanks our speaker. Thank you, ma'am. We are very
Thank you.
grateful for the time and effort you took to share your thought and experience with our audience.
Yes. No,
Our
thanks. Thank you very much for this invitation. And I would love to be in association with your society and your efforts. So whatever can be done from my side, just let me know. Thank you.
Sure, ma'am. Hopefully in future, physically, we organize some event and we will come to
Perfect.
IIT Indore.
Yeah. Yes. Okay. Thank you.
Thank you so much, ma'am.
Bye.
Bye.